So good afternoon all and welcome to our webinar in collaboration with PharmaLex. On behalf of One Nucleus, thank you very much for joining us this afternoon. Our speaker for today's session is Dr. Bruno Boulanger, who is Global Head of Statistics and Data Science at PharmaLex, Belgium, and also CSO and co-founder of Arlenda. So Bruno has more than 25 years experience in pharma R&D, and as well as numerous industry positions, Bruno also has academic credentials as a lecturer at the University of Liège. And he's also co-authored more than 100 plus publications. So the objectives for today's session include an overview of the current trends in validation of analytical procedures and bioassays, an update on PharmaLex software solutions, as well as some guidance on upcoming changes and how the software solutions can help you to stay compliant for now and in the future. Before handing over to Bruno, just to clarify on some of the housekeeping. So we are recording the session with the aim of using that to send out to other registered delegates who perhaps didn't join us sort of after the session. And in case you missed anything and you want to revisit some of the key takeaways and learnings, you'll receive a copy of that recording too. The chat box is enabled, so throughout the session, I'd be grateful if you could stay muted, but then, please don't hesitate to ask a question and we will pick those up after Bruno's presentation. So we're aiming for roughly 40 to 45 minutes of presentation and then the remaining time for Q&A at the end. So at that point, if you'd like to unmute to ask your question, that's also very welcome. So we can make this an interactive session as well. So with that, I will hand over to Bruno now, and I do hope that you find it a useful and informative session. Many thanks, and over to you, Bruno.
Okay, thank you, Alicia, for the very nice introduction, and thank you to all participants to join us for this afternoon meeting. So I will assume that you can see my slide.
Yes, we can see your slides.
Yeah.
Yep.
So today we are going to discuss about the very important topic of validation of analytical procedure or bioassays, and I'm going to give you some perspective about the current trends and current thinking and what we may expect in the near future. Because a lot have changed. So this is the agenda of the day. I'm going to discuss about, I will say, all the current guidance, discussion, debates that did happen over the last, I would say, 5 to 10 years, because to revisit, to rethink, and to re-examine the way we conduct validation of analytical procedure and bioassay since the ICH Q2 first version that has been released. And then I will show you and I will give you some insight why the software that has been developed at PharmaLex are already compliant not only with the recent text and thinking, but I'm also giving you an idea of what we may expect given the current discussion that are happening. So just to give a big picture about where we are, the challenge we are facing and the regulatory authorities everywhere a little bit are facing is how to bring together different type of approach. There is a lot of debate and discussion, for example, within the US Pharmacopoeia about the life cycle, the fit for purpose, the analytical target product profile. There is the quality by design, design space, and life cycle approach regarding the process Q8, Q9, Q12 regulation. There is the ICH Q2 regulation that you know, and there is also the international standard organization and with the concept of uncertainty. And now there are recent documents that are coming and that have been released, and that USP try to also give some feedback. And so the idea will be to in all those approach, there is something that makes sense, which is meaningful, that need to be combined together. Okay, so just to recap the big picture, the life cycle approach, and not only the validation, that is really the trend that is going to happen in the near future, is to see all the life cycle of a method. How we develop an analytical method, or we validate an analytical method, or we make a transfer or bridging, and how we are going to have a control strategy and potentially improve the method and so on. So ICHQ2 was about the validation, simply, but now the industry is moving toward a full global vision, and that makes a lot of sense. And now we will need to see how we need to envisage the validation given the big picture in the future. So ICHQ2 revision and ICHQ14 are, according to the ICH website, expected by the end of 2021. And as you can see, the objective of those two documents is about the development, stage one, I have to say, how to develop analytical method, and all those development approaches going to help the future. But as you can see, risk-based thinking, and as well a change management. So it's really trying to embrace the life cycle approach. The Q2 revision, it's certainly going to enlarge and include many concepts. Why? Because there are many new technologies coming on board that are becoming more regular, like spectroscopy, near-infrared, Raman, and so on, that need to be integrated. It's not only about adding few lines for those new technology, but the fact that those new technologies are coming on board, it will require to envisage some of the criteria of validation in a slightly different manner also. And those two documents are likely to be discussed not independently, I hope, but together, so that Q2 will also be able to cover the needs of the life cycle approach. So what we have seen in the recent year, and some publication by the stimuli paper by the USP, you can see some of the new concepts that are really becoming the reality. Is about life cycle. It's about analytical target profile, which is a concept coming from the QBD type of approach. Concept like uncertainty, which is something that did not exist up to now, and that is going to be existing in the near future, and what we call the target measurement uncertainty. Yes, still part of piece of this life cycle approach. You can see there is the analytical control strategy, how to make a decision, fitness for use based on uncertainty, and how to discuss about the reportable result and the uncertainty. So as you can see, these are new words that are becoming, that are be used in many official documents more and more, like ATP, TMU, uncertainty, and reportable value. And the objective right now is to give an idea how those pieces all connect together and what do they mean exactly. So continuing the new general chapter of USP, the 1225. As you can see, it's about life cycle management, trend analysis, continually assure, periodically review, and so on. So you can see that the whole concept of validation is not a snapshot picture in the time, it's really seeing the big picture from the development to the control strategy. Again, that's a new document is always the same idea. And you can see something from the USP consistent with the concept of quality by design, as described by ICH Q8, Q9, Q10, and Q11. So you can see all the concepts that are existing on the process side, in fact, do fully apply to the analytical procedure. And therefore, the same thinking, the same of approach is going to be part of the future. The FDA release in 2015 is guidance. And as you can see in this guidance, there is this life cycle management, revalidation, comparability, transfer, and so on. So these are all concepts that are coming and that are already since a while integrated in our suite of software to do the job, validation and transfer. Okay, I continue and I dive more and more in some of the detail. In the USP 1210, you can see some new ID coming. For example, the concept of linearity that has been a lot of confusion in the past and misunderstanding is becoming more and more clear in the future, in the sense that, for example, you can see linearity may be inferred from accuracy, and we will show how we do that, and the concept of tolerance interval, of prediction interval that are coming into the game, and I will explain why it is really needed in the near future. Okay, so looking at 1210, and 1210 is really giving clear indication about what you may expect in the near future. Is validation must provide evidence of a procedure about the uncertainty, statistical intervals, the reportable value and concept, and something which is very important here is that establish a single criterion that can be simultaneously validate both accuracy and precision, and I will add and linearity and limit of quantitation and so on. Okay, that's important, this concept of single criterion, because what is currently right now, there is a number of acceptance criteria like accuracy, precision, intermediate precision, repeatability, and so on. And usually the practice is to define acceptance criteria for each of them. But we will see that it doesn't work, because it doesn't work very well. We will see that it's very important. We make one decision. Is it valid? Yes, no. So we need to have one criteria that captures the whole performance and future capability of an assay to make a decision. It's there. I mean, it's just a way to assemble the different pieces in a meaningful manner. And this has been proposed in a series of papers already that has become famous since, and it's there. It is the concept of, we will see, total error and accuracy profile. Yeah, just to mention that those papers that are now 20 years old have already been integrated into Innoval and Silva, the suite, since a while. Yeah, still in 1210, for the limit of quantitation, the important consideration is the acceptable precision and accuracy. And you see, there is already a debate about the fact that we need to evaluate the precision and accuracy together, but it needs to go beyond this. So these are, I would say, a number of important documents that are existing since a while, but that has been recently updated, changed, improved, and there are even new documents in analytical procedure. You see, many of those documents have been reviewed. Those one has been reviewed, and there are new documents from FDA, EMEA that are coming, and more to come in the close future. So this is the background, so you can have a feeling about what may happen in the future. There is this life cycle approach, which is something very important, from development to control strategy. And the fact, and the second piece is that we need to have one criteria to grasp in one shot what is the future capability of the analytical procedure. So I'm going to give you a big picture about this. So the objective of an analytical procedure is to ensure it will be able to quantify closely each of the future unknown sample. So meaning is that for each reportable value need to be very close from the unknown true content of a sample. It need to be in the neighborhood, as I will say. So, but what do we mean by close enough and any future with a high probability? The idea is that the reportable value need to be close from the true value. But we need to be sure it will be close within 2%, within 10 milligram. We need to define an acceptable range. That is the maximum distance or difference between the observed, the reportable value and the true value. But keep in mind that when we are using in routine, we cannot compute, we cannot evaluate this distance between the reportable value and the unknown value because it is by definition unknown. So the only thing that we can do is to say the probability that the reportable value will be close from the true value is very high. So during the validation, we work with known sample, and we can evaluate how far the reportable value is from the true known value. But that's not the purpose. The purpose is to evaluate in the future unknown sample. Okay, so the only piece that we can have regarding the future unknown sample is to say there is a high probability that my future measurement will be close from the true value. That's the only piece we can say. Okay, and this probability, in a way, is defining, or risk, if you want, is defining the quality, the overall quality of your method. That's one way to summarize this. Okay, so going back to some statistics, what you can do is to say, assume that you have the lower and upper specification limit, which are your acceptance limit for the reportable value, for the result. Okay, you can say all my results need to fall within plus or minus 2%, when it's plus or minus 5 milligram, for example. Well, if you know the distribution, if you know the distribution, what you can do, you can compute what's the probability to fall within the acceptance limit, which is the gray area. And you can say, well, I have 80 or 90% chance to fall within the acceptance limit, and therefore I can trust my method. Oops, sorry, I'm going the wrong way. But there is an issue. The challenge is the following, is that you don't know what is the bias of your method. The bias being the difference between the true value and the mean of several measurements. And you don't know what the precision of your method. Well, you can do a number of experiments during the validation, but it will just be an estimate of bias or estimate of the precision. If you repeat the same validation experiment, you will get a different bias and you will get a different precision, slightly different. So that's the whole idea, is that you need to take into account also in your probability of success, you need to take into account that you are not totally sure about the bias. You have a good idea, but you are not perfectly sure. And the same for the precision. So don't think that the bias and the precision you observe today is going to apply tomorrow. It gives you an idea, but it is not, it's just an estimate and there is some uncertainty. So what is happening is that you are going to say, the objective of the validation is not to ensure that you know what will be what we call the predictive probability in the future to be close from the true value, given the estimate of the bias and the precision you observe during the validation. Okay, so in this case, what is happening is that the performance estimate mainly mean bias and precision, repeatability, intermediate precision, is not the objective. It's just an intermediate piece to be able to evaluate if in the future, with unknown sample, the analytical procedure or the bioassay will do the job. Okay, And this is what we call, and this is what you can see in the current USP document, the predictive distribution. What can be the accuracy in the future, given my current estimate? And so that's the idea. Assume that you have two analytical procedures. You can see that, for example, from the validation, this analytical procedure is giving reportable value with some level of precision, but no bias. And given this, you can say this is the probability in blue here to be out of specification, which is the risk. You have another analytical procedure with a much better precision, but with a bias, because you see it's a bit off. But the risk, which is the total blue area, is the same. So you can see that you may have totally different bias and precision, but still having the same risk. The risk being defined, probability to be out of specification. Okay, so that's the risk that matter for the future decision, not the bias and the precision, because the bias and the precision doesn't tell you anything about the quality of one reportable value. And you can see here, I take this example. This is the difference between one reportable value and the true value. This is what we call the accuracy. And this is the same. So I can have the same accuracy with two different type of analytical method. So the idea is the following, is to use the what we call the predictive distribution or the predictive interval to compute the probability in the future, given the result from the validation, to be within the specification, within not being too far from the true value. Okay, so sorry, it's a busy slide here, but what is very important now, and that's where I start to connect the different pieces together, is that the predictive distribution that we compute classically in statistics is in fact equal to the uncertainty of measurement that we can observe in the ISO regulation. It is the same. Not the same idea, it is mathematically the same. So this predictive distribution on the uncertainty integrate three pieces of information: the precision of the method, the uncertainty about the bias estimate, because we make an error when we estimate the bias, the uncertainty about the precision estimate, because we make an error when we estimate the precision. And so the uncertainty is taking all those three pieces all together, okay? But the predictive distribution doesn't integrate the bias estimate. And that's one piece, but we will see where it is coming, where it will be included. And the uncertainty, there is frequently this confusion which is made in the literature. The uncertainty of measurement is not the precision. These are totally two different concepts. Okay. But if we compute the probability, as you can see here, using the predictive distribution to be within the limit, this probability to be within limit, acceptance limit for reportable value, integrate the concept of bias. So what it means that if you work with the predictive distribution, you have the accuracy. And if you have the uncertainty, sorry. And if you combine this concept of uncertainty with the target measurement uncertainty, your objective, your acceptance limit, then you integrate all those pieces of information together. And that's the way you can connect the concept of bias, precision, total error, uncertainty, risk through one equation. And I will show you how it works. So just a quick reminder is that you see here it is a sketch of an analytical procedure. This is the true value. The difference between one reportable value and the true value, which is what we call the accuracy. Okay, and the accuracy is not the difference between the true value and the mean of several measurements. Just to remind the ICHQ2 definition, the accuracy is the closest agreement between the value, one value, which is accepted as the true value, and the value found, i.e. the reportable value. Okay, and you can see this in some text, some regulation indicating, okay, the term measurement accuracy should not be used for measurement trueness. Well, the whole idea is to say, be careful, accuracy is not the bias. It is the agreement between a measured quantity and the true quantity, which is not the bias. Okay. So, yes, so USP 1210 and ICHQ2, Q2 say, well, the bias and the precision and their confidence limit should be reported. But okay, that's just a way to evaluate this. But be conscious that bias and precision is not part objective. The objective of the analytical procedure, you would like to be sure that in the future you can trust a reportable value. Okay, you can trust a result. But just to mention that a result is accurate. But the procedure has a bias and a precision. Of course, you need to have an idea about what the bias and precision to figure out if the result will be accurate. Okay, so do not confound the bias and precision, which are properties of the procedure or bioassay, and the accuracy, which is the quality of one reportable value. These are different concepts. Okay, and that's the reason, as we have seen, we have to use the prediction interval. And the way to summarize everything is to use the probability of having accurate reportable result in the future. So the way it works graphically, and just to give a feeling about the way, for example, our software are doing, from the validation result and experiment, what you can compute over the range that has been investigated, you can compute, like we can see in blue here, the prediction interval. And you can see that over this range, the prediction interval is within the acceptance limit, which are -15 to +15, that we can call the target measurement uncertainty. The width of the prediction interval here represents the uncertainty of the measurement. And the red line represents the estimated bias over the range. And as you can see, this graphic is telling you the probability there is 95% chance that future measurement will be within ±15%, which was my objective, my target measurement uncertainty. Therefore, you can declare that over the range, the analytical procedure is able to do the job. But now there is an additional challenge that where there is sometimes misunderstanding about how to combine uncertainty into the decision. As you can see, and as we can see here, the uncertainty is not the same over the range, of course. Precision is not the same either. And therefore, I present a sketch here about what's happening below. You have small uncertainty, and the uncertainty is becoming larger for the high level. But the problem is that when you work with an unknown sample, you don't know at which level you are, because the level that has been used here are arbitrary level covering a range that has been decided during the validation. But with a known sample, you can be somewhere in between. So given you are in between here, at a level that has never been tested into the validation, you don't know what's the uncertainty. Are you here or are you there? Or you probably are in the middle, but you don't know how much. And that's where the challenge is becoming. And the reason this graphic is very important, because it's telling you, well, there is a lot of chance that if I have something around 200, for example, here, at a level that never had been experimented in the past, well, I have the feeling that I have a high probability to be within the specification. Nothing dramatic will happen between those two levels. Probably it's going to be reasonably smooth. So that's another way. Once you have this is the previous graphics, you can also compute the risk. What is over the range, the probability I will be out of specification. So it means you see here the probability is usually below 5%. So what it means, tomorrow when you have a reportable value, okay, you can claim that there is 95% chance that the true value is within plus or minus 15% of your reportable value. You're sure about that, okay? You cannot evaluate this because it is an unknown sample, but you can claim there is 95% chance my true value is in the neighborhood being defined by your acceptance limit. Okay, that's very important. And so that gives you an idea about the risk you may take over that range. You see this risk is very low, therefore you're confident about claiming that. This risk integrates all the aspects: bias, precision, uncertainty and bias, uncertainty and precision over the range. Okay, now the extra benefit from this is that you can also use this. Assume I have an ELISA assay that requires either a 4-parameter logistic function or a 5-parameter logistic function, as you can see here. The question, which one is I have to use? When you take a look at the quality of the fit, well, the quality of the fit is pretty good for both of them. But the objective is not the quality of the fit, because I don't want to reduce the error in the response level. I want to reduce the error in the inverse prediction, i.e. on the concentration level. And so if you are using this calibration curve, this is the accuracy profile you have. You see in blue my prediction, in black my acceptance limit, in red my bias estimate. If you use this curve, you have this accuracy profile. I know the question, which one do you prefer? Okay, because you can see over the range, they give you something which is pretty good. But if you have a close look, you can see that with the 4PL, okay, you can have a shorter limit, a smaller limit of quantitation. Here, the limit of quantitation is about 25, and here, the limit of quantitation is about 10. Okay, remember, limit of quantitation, bias, and precision should be acceptable. So that's interesting, because it means that if you are looking to improve your limit of quantitation, expanding your range, this method, this 5-parameter logistic is giving you a better result. If you are not worrying about decreasing, giving a small limit of quantitation, well, the 4-parameter is doing a great job and is easier to use. So you can see both of them are having a good fit, but one is allow you to achieve another objective, which is having a lower limit of quantitation. So that's one benefit of using the accuracy profile. You can evaluate the quality of your calibration curve. The other benefit is that if we move into a life cycle vision, which is to not only to design, assess, but also to have a control strategy, as it is going to happen, this accuracy profile is giving you also, and this prediction interval is giving you also an interesting value. Let me explain. Here is a real, here is two procedures that has been used, two HPLC procedure, in fact, dosing the same product and at the same concentration and so on. That has been done in parallel. During the validation, with the procedure 1, we got the blue interval here, the blue line, which is the prediction interval. And this one is much better than this one, as you can see. So by default, you will expect that procedure 2 doing a better job than procedure 1. But nevertheless, it has been done in parallel. And those limit of those prediction interval has been used in routine as, that's the red lines here, has been used as the control limit later on. So here, during the validation, at this stage, you predict that 95% or 90% of your measurement will be within those two limits. You use those prediction interval as your control limit. And then it allows you to assess that you should observe 90–95% of the control sample between those limits. Okay, and it works very well. You can see that in routine, this is what we can observe. If we predict a large variability, this is what we observe when we use the method in routine. If we get a narrow variability, you can see we are pretty consistent. And it allows you to detect. So the benefit from this is that not only you can make a decision about is my method able to be able to do the job, but you have an extra benefit. Those prediction intervals are becoming your control limit to ensure that you control the risk at the same level over time, which is also something very important. You don't want to have too much false positive and false negative in the future. Prediction interval will do the job correctly and will detect the problem, but will not detect too often issue if your method is doing a great job. Okay, so in summary, what you can see is that with this concept of combining bias, precision into a prediction interval over the range and computing a probability, allow you to make a decision using one single criterion, probability over the range to be within the limit, and also give you a good idea on the way to develop a control strategy that will control the risk over the long run, control and even drift you may observe in the future. Okay, so these are all the software that we have developed at PharmaLex since a while right now, which are Innoval, dedicated to physicochemical methods like HPLC, Silva for ligand binding assay and even relative potency assay, and Transval for the transfer of analytical method, because usually we develop a method in one lab, we transfer to another lab, and we need to ensure that the result will remain consistent. What is the value of those software? Well, it's not only a statistical software, it is what I will call a productivity software, because it automatically generates a validation report, which is ECTD compliant, but also compliant with all the regulatory documents you have, ICH, Q2, FDA, and so on. Okay, the software is fully validated according to 21 CFR Part 11. Yes, it's a software as a services. You can access it through the web. You don't need to make any particular install on site. It's pretty safe, user-friendly. I will make a demo right now and focus on decision making. Okay, so these are all the criteria you can see in Innoval. We have Silva. We are dedicated for bioassay and relative potency assay. And if you go back into 1033 in USP, biological assay validation, if you read this, all the requirements are existing and have been implemented in Silva. Okay. And Transval is for the transfer. So I'm going to rapidly make one demo. On one software. Yeah. Okay. So if you go on the PharmaLex, SmartFlex website from PharmaLex, you see statistic here, and you have our software for validation, Silva, Transval, and for stability also. That's another debate. So If you click on that one, okay, yeah. So it's giving you a lot of information about Smart Innoval, or Innoval, how it works, give you a number of characteristics. You can request a demo and have a demo version, or you can log in directly into the application. And this is what I'm going to do. And I will not go through all the menu, but I'm going to do a validation. And the data I'm going to provide are the data where I have the response that I need to calibrate. And having this calibration, I need to compute the inverse prediction and so on. Some case you may have directly the inverse prediction available because your analytical device is giving you directly the result. Here I do the response as well. So now what you need is to enter the data, and you can enter the data manually if you want, but of course the best way is to use you can have the data organized in an Excel sheet, and I will show you the way it has to be organized. Okay, so there is information about the project, about the unit and so on. Everything is in the file I did upload. And here I have two sets of data, the one for the calibration data with the concentration and the signal, and the validation standard data with the concentration and the signal I obtain. Okay. So the data uploaded on the website, and then I need to define my limit. The famous lambda in my presentation, I need to say, yes, my criteria is plus or minus 15%, and my risk level is, for example, 10%. I don't want to have more than 10% out of specification for my method. You can change to whatever level you want. And these are all the great regressions that are envisaged in this case. Now, the software now is going to compute all the regression model, all the inverse prediction, everything is going to be computed all together, okay, for all scenarios. Okay, and then what you get back is by model, you have an idea about the accuracy, the range, the precision, the trueness, and the limit of quantitation, lower and upper. And the model are ranked from the best to the worst. There is a criteria. Criteria range between 0 and 1. And then you can say, well, my linear regression is doing a job which is as good as a weighted linear regression, so I'm going to keep that one. But in my report, I may also need to show an alternative option to justify the choice of the linear regression. Okay, now I'm going to generate a report. And yeah. I'm going to generate the report. The first thing I have is I have a report in HTML format, so I can review, and I can even potentially add comments. And once I've made it, now I'm generating my ECTD compliant report. That is already a full validation report. You know, I have on my table of content. This is my response function. You can see this is my calibration. I have the R-square, for example. This is the trueness or the bias. And I have the recovery, the confidence interval recovery. I have the precision. And as you can see, all the formulas are in the report. The report are standalone. So everybody can review and check. There is nothing black box there. You have an idea about the repeatability, intermediate precision, and the confidence limit. You have a table about the uncertainty of the measurement, which is very important. And then you have the accuracy, the famous accuracy profile I was showing previously. Okay. With the corresponding risk profile, which is the probability to be out of specification. And there is, of course, as mentioned in USP 1010, the linearity can be derived from the accuracy. This is the unit line that you introduce concentration, and this is the back calculated concentration on the y-axis. And you can see this is very well aligned. And probably there is a R square that has been reported somewhere. Okay. So you got everything, you got all the reference that has been used here, all the data that has been introduced are available. And then you have also the alternative. You can see that the accuracy profile with the weighted regression is not doing as a job as great as the previous one, because I'm about I have a problem of bias probably here. And the other regression, which is the linear with another weighting strategy, you see it's really not doing a great job, because on the lower hand, I'm out of my accuracy and specification. And that justifies I choose the simple linear model. And then you have all extra information in the appendix. Okay. But the key point is this one is you have one assessment. This is my risk for future unknown sample. And you accept the method if the risk is small, which is really the future philosophy of the text and the life cycle approach. Okay. So I thank you for your attention, and now I will give the time for a few interactive questions you may have. So thank you for your attention.
Thank you very much indeed, Bruno, for that expertly delivered presentation there. As Bruno said, there is some time left. In case anyone on the line has any scenarios of their own, they want to use this time to discuss them directly with Bruno, it's a brilliant opportunity to do that. So by all means, you can use the raise hand function, which you can find if you click reactions on the bottom toolbar, or you can put something into the chat box, or feel free to just unmute if that's easier. But if there's no immediate questions, Bruno, just to clarify, what would be required to set the software up at a typical lab site?
Okay, in fact, very little, because as you have seen, it is software as a services. It is web-based at this stage. So you need to have an access to internet, at least in the computer. But we also provide a document for the qualification of your software on your side, meaning that we send you sample of data to use, reports that we get, and you run all those examples, all those data at your site, and you need to check that what you get, the report you get is the same as the report we generated. And this proves that indeed it works correctly, transfer of data, maybe you have a version of Adobe which is not the same, that may mess up with the graphic whatsoever. Everything can happen. But we provide you the document to fully qualify on your side. But the only thing you need is to have an access to internet and to have an account. Just drop an email. If you want to have a demo, you ask to have a demo account, and we will provide you with an account and a password.
Thank you. And so I guess then if it's web-based, if there's companies that are doing remote monitoring of experiments, then that's possible as well?
We don't do any monitoring. Okay.
Sorry, if your experiment is remotely based.
Yes, that's okay. Sorry. Yeah, and that's the value, and that's a great interest. And the reason it interests a lot of companies is that, for example, you may have a company that is asking to a contract lab to perform the validation and the development. Okay, but so instead of asking to the contract lab, I would like you to buy and to purchase the software. Okay, the company can share and say, well, here is the link, here is an account, and you simply upload your data that way into the software. And so you ensure that the sponsor company is going to receive the report correctly done with their standards, but without embarrassing the contract lab about using software agreement and so on. It can be shared. You have a contract lab in India, just send the link and that's it.
And absolutely, and in this kind of new world we're operating in, where things are done much more online, I'd say it's very,
and it's becoming the practice, the common practice today. Yeah, to do
very
that.
timely.
Mm-hmm.
So just to double check then from the audience, there's no it's a brilliant opportunity to speak directly with an expert. So please do take it if you're. I assume we have people on the line performing their own assays. So if maybe there's a scenario you wanted to discuss or an example, it could actually enlighten others on the call as well. Or just feel free to let us know in the chat box if you found the content useful. That would be very much appreciated. Thank you. And so finally, Bruno, so in terms of just a final question from me, so what are the options for validating the installation of the software? Are they similar between the different types, or do they differ?
Between the different software?
Yeah. Mm-hmm.
Yeah, there is a validation package that we provide, which are different from software to software.
Okay.
Yes, indeed.
Yeah.
Yes.
So depending on which is selected, you'll receive all the necessary kind of.
Yes, exactly.
Yeah.
But also it comes, I'm just showing the software, but what is also available directly on the website are user's guide, demo version, examples, and there is a lot of, and of course, there is a follow-up on this in the sense that if you have any question, something you do not understand, we work remotely, drop an email, and we set up a remote call to explain some detail how it works and so on. Okay. The beauty of having something which is central is that we can work remotely wherever you are and helping you in real time. But we provide validation package so that you ensure that the installation and the way it works at your site, given your your computer, your internet, the version of everything is going to work properly.
Excellent. Thank you very much. So just to recap, I will send a recording of the session to all those registered. So if there was any part of the presentation perhaps you wanted to revisit for some of the technical detail, then you can do that. And equally, if you leave the webinar and you decide actually you would like to explore the solutions further that have been presented and how they may help you in your own laboratories, then of course I'm more than happy to join any dots. So you can get in touch with me, and then I can put you in touch with PharmaLex. So I would encourage anyone on the line to get in touch if you would like to explore any follow-ups. So with that, I will probably close our session today. And thank you again, Bruno, for your time, for the excellent presentation, and also for the demo. That was a bonus at the end to actually see some of the software in action and sort of the outputs, what that looks like, and the usability as well. So that was really interesting. Thank you. And to all those on the line, thank you for joining. Like I said, don't hesitate to get in touch if you do have any questions after the session, which you perhaps didn't think of now, and I'll be happy to help. I'll put you in touch with Bruno or colleagues. So with that, I just wish you a good afternoon and say thank you for joining. Thanks.
Okay, thank you. It was a real pleasure. Yes.